Friday? Survived one day of biochemistry? Think about that. You've already survived one whole day of biochemistry. But then so did I. I'm not sure what that means. So, welcome back. Let me pull up the other screen here so that we get all of them showing. And that should bring it up. Okay. Fancy background. I spent a lot of time working on that background of that one right there. Hope you guys like it. Okay, so hopefully we got all the business straightened out. Everybody got emails from me? Everybody's gotten the links to the YouTubes and that's working okay? They weren't originally. They are now. I think they're all there. So if you haven't looked in the past 12 hours or so, take a look. They should be working fine. So hopefully that's the last of the glitches. Anxious to dive into stuff. So today I'm going to talk about some basic review for chemistry. Just a very tiny bit of organic chemistry, a little bit of freshman chemistry, and expanding on that freshman chemistry with respect to buffers and weak acids and so forth, because that will be very relevant for us as we move into next week talking about amino acids, charges of proteins, and things like that. So it's important that you understand ionization. Okay? Ionization, very, very critical for you to understand, because if you don't understand ionization, you're not going to understand what I'll talk about next week and some of the problems that will be on the examination. Okay? All right. So let's dive into that. Our first topic I want to talk about is just sort of a review of something I assume everybody knows. Everybody in here presumably has had organic chemistry, or they've been warned by me that they should have had organic chemistry. The various groups that you see on the screen, we all hopefully know what alkenes, alkanes, alcohols, ethers, amines, all of these things are. So I'm not going to go through and in general define those for you in the future. But those are groups that we'll talk about a lot. We'll talk a lot about carboxyl groups especially in the first few days. We're going to talk a lot about amine groups or amino groups as it's listed up there because, of course, carboxyl groups and amino groups are important constituents of amino acids, and since amino acids are important constituents of proteins, we need to understand those. Okay? We're going to see what's going to happen over the next couple of weeks is that I'm going to try to impress upon you the real importance of structure and function. Structure and function is where everything is at in biochemistry. If you understand, you can't understand function unless you understand structure. So we spend the first part of the term getting all consistent on structure. Structure is affected by a variety of things in proteins, and proteins are really where it's at as far as cells are concerned. Structure is affected by charge. Structure is affected by hydrophobicity. Structure is affected by covalent bonds. Structure is affected by ionic bonds, hydrogen bonds, a lot of different things. So it's important that we all get up to speed and understand those things to start. Okay, I've lost control. Okay. Ooh, come on, don't do that to me. There we go. Okay, so we all learned from inorganic chemistry, of course, that carbon is the central, most important atom in organic chemistry. We learned that carbon has orbitals that hybridize to produce a tetrahedral structure when we have 4 different orbitals. And what you see depicted on the screen is a molecule of methane in which the carbon in blue is bonded to 4 different hydrogen atoms. And that structure that results from that is known as a tetrahedral. The tetrahedral structure is a 3-dimensional structure. And we remember, hopefully, again from organic chemistry, that whenever we have a carbon atom that has 4 different things attached to it, that the 4 different things can be attached in 2 different ways in 3 dimensions. We refer to these as handedness. Okay? Biochemists use this term a lot. Organic chemists like RNS structures and so forth. And biochemists are pretty stupid people. So we talk about handedness, left-handed, right-handed. And I think you'll like what we do with stereochemistry and biochemistry because we try to really keep it simple. Again, we're simple people, biochemists are. All right. There is a depiction of 2 different molecules, one that's in a left-handed form, one that's in a right-handed form. You can see that they are mirror images of each other. And we know that they're handed, that is, they have different three-dimensional structures because the carbon has four different things attached to it. It has a hydrogen, it has a hydroxyl, it has a methyl, and it has a carboxyl group. Okay? So you see the depiction in three-dimensional space of the two different ways in which those four groups can be arranged. It takes four different groups to have handedness. If you have only three different groups, let's say I had two hydrogens up there, then I wouldn't have handedness. Now, handedness turns out to be really important when we talk about amino acids and also when we talk about sugars and carbohydrates. Okay? There's a bias. There's a strong bias with respect to the handedness of amino acids, as we shall see. Another thing we need to remember from our freshman chemistry, this is from our freshman chemistry, not our organic chemistry, but from our freshman chemistry, we need to remember electronegativity. It's one of the first things I ever learned in freshman chemistry, and probably is one of the first things that you learned as well. Electronegativity is a measure of the affinity or attractiveness that a nucleus has for electrons. I'll repeat that. Electronegativity is a measure of the affinity or attractiveness that a nucleus has for electrons. I'm hoping you remember from freshman chemistry that those atoms on the left-hand side of the periodic table had a very low electronegativity, a very low affinity for their electrons. They were happy to let them go. And those on the right side of the table had a very high affinity for electrons and really pulled them close. Well, that's really great if we're talking about sodium and chloride. Sodium, of course, giving up an electron very readily, chloride taking it very readily, making salt, forming ionic bonds, a positively charged sodium and a negatively charged chloride. In organic chemistry and in biochemistry, we don't have such divergent electronegativities, but we do have different electronegativities. Okay? So the different electronegativities that we have are actually shown on the screen here. If we compare, for example, the electronegativity of oxygen, higher electronegativity means greater affinity, stronger pull on electrons. Lower electronegativities means less affinity, less pull on those electrons. If we compare oxygen to hydrogen, we see that there's a pretty fair difference in the electronegativity of the two. And so if we think about what's happening in water, where we have a hydrogen bonded to an oxygen, we actually have two hydrogens bonded to oxygen, of course. Each oxygen is going to pull that electron a little bit away from hydrogen. Doesn't make an ionic bond, but it does pull that electron closer to oxygen than it is found to hydrogen. Now that becomes important for things we'll talk about hydrogen bonds. We see there's differences between how carbon and oxygen are. It's a pretty fair difference between carbon and oxygen, for example. And all of these things will contribute to the structure and the attractions that we see within and between biomolecules. So hydrogen bonds, very, very important. And those hydrogen bonds arise as a result of differences in electronegativity. I'll give you a couple of examples in the lecture today. Here's one. We see in this structure a water molecule in the middle, and it's surrounded by four other water molecules, and you'll see a bunch of labels on there. The oxygen in this case is shown in red. The hydrogens are shown in white. And we see that hydrogen bonds are labeled. Those hydrogen bonds are attractions that are occurring between the hydrogen of one of those water molecules and the oxygen of another. And that's important because that's a force. That's an attractive force. Well, how does that arise? Well, if we have a difference in electronegativities, which is what we have with oxygen and hydrogen, that difference in electronegativities gives rise to what we call a partial charge, not a full charge. Full charge happens when you completely gain an electron or you completely give up an electron, but a partial charge arises when you don't equally share those electrons. The big kid taking the lunch money from the little kid, right? Well, I guess in that case, it's pretty much a take money away. It's not like I'm going to share it with you. But they're going to have a tug of war, and the big kid's going to hold that money closer to them than the little kid's going to get to, right? That's what's happening here. So oxygen ends up being partially negatively charged because it is pulling electrons closer to it. Hydrogen is partially positively charged because there are less electrons associated with it, right? So those partial charges, partial plus and partial minus, are attractive, just like minus and plus are attractive. I want to remind you of something that I find shockingly, and I don't blame students for this, I blame our instructional system for this, and this is also true for the electronegativities, that students lose sight or didn't maybe get taught as well what reduction and oxidation is. We're going to talk about reduction and oxidation a fair amount, especially as we get into talking about metabolism. Reduction, I want you to remember, is the gain of electrons. A molecule gets reduced when it gains electrons. An oxidation occurs when a molecule loses electrons. They go hand in hand. If a molecule gets oxidized, something else is getting reduced because those electrons that leave one molecule go to another molecule. For every oxidation, we have a reduction. Okay? Now we'll see that it becomes really important when we get into biological systems where those electrons actually get transferred to electron carriers. And that's kind of a cool thing that happens biologically. But remember that reduction is the gain of electrons, oxidation is the loss of electrons. This reaction shown on the screen depicts the reduction that's happening of acetaldehyde. Acetaldehyde is the molecule on the far left. Acetaldehyde, we'll talk later, is the thing you can know as Mr. Hangover. Okay? In the reaction that happens in your body after you've had a few beers, you've had a few alcoholic drinks, what happens is alcohol, which is the molecule, well, what did I do? I hate presentation software for this reason. Okay. The molecule that's shown on the right, this guy right here, boo, 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 boo. Another reason I hate presentation software. On the right side of the equation, H3CH2COH, that's ethanol. That's what you're getting in your beer. In the reaction that happens in our body, that reaction is moving to the left. You're making acetaldehyde. Your liver's doing that. And that acetaldehyde gives you a hangover. Okay? This reaction is shown in the other direction. We're actually reducing acetaldehyde. We see the aldehyde group becoming an alcohol group. That's a reduction. You should remember from organic chemistry. The electrons for that reduction are coming from the molecule called NADH. And NADH is giving up its electrons, donating those electrons to the aldehyde group. That's how the aldehyde group is becoming the alcohol group. Protons come in, as you see here. And you see NADH is getting oxidized. NADH becomes NAD. So we have a pair, reduction, oxidation. Acetaldehyde is the oxidized form. NADH is the reduced form. Ethanol is the reduced form. NAD plus is the oxidized form. So we've seen the oxidized has become the reduced has become the oxidized. So there's been a swapping of electrons is all that's happened. And we'll see a lot of this biologically. Okay, Gibbs free energy. There's freshman chemistry summarized in one slide for you right there. Okay? Freshman chemistry. All right. Probably you didn't like Gibbs free energy when we talked about it in freshman chemistry. All right? Gibbs free energy is such a useful concept. And it becomes very important for us when we talk about biochemical reactions, because what Gibbs free energy tells us is the direction of a reaction. And when we talk about biological molecules, the direction of a reaction is very important because we remember, first of all, that all reactions are reversible. And when you talk about metabolism, which are the reactions that's occurring inside of your cells, we can break metabolism up into two components: catabolism and anabolism. Catabolism being the breakdown of molecules, anabolism being the building up of molecules. And guess what? Whether we're doing catabolic processes or anabolic processes is simply a definition in terms of the direction that we're headed. So if we understand direction, we have a better understanding about whether we have a breakdown process occurring or a synthesis process. Breakdown being catabolism, synthesis being anabolism. Well, Gibbs free energy, a lot of equations. I try to spare you as much as I can in terms of equations. And by the way, I have a deal with you guys. You don't have to memorize any equations in this class. Zero. Okay? I'll give you every equation you need on the first exam. Yay, there you go. Okay? No equations you have to memorize. So nothing up there that you have to memorize. In fact, I put the hardest equations up there because I like to keep Gibbs free energy equations very simple. What we're really interested in is the overall Gibbs free energy positive or is the overall Gibbs free energy negative? We'll talk more about that later in the term. But we can see on the screen the Gibbs free energy equation is the second from the bottom row. We see the delta G, that's the change in Gibbs free energy, is equal to a quantity we call the delta G0 prime, which is the standard Gibbs free energy, plus the gas constant times the temperature in Kelvin times the natural log of all that stuff that's up there. And I'll simplify that for you later. I'm not asking you to memorize that right now. But I want you to remember that the Gibbs free energy is, in fact, very important for us to determine the direction of a reaction, and that the direction of a reaction can be controlled by cells by controlling the concentration of products and reactants. That's the only control that cells get to have about directions of reactions, the concentrations of products and reactants. Cells are control freaks, as I like to say, but the only control they have is that right there, and we'll talk about that later in the term. Well, here's water again. I want to just say a little bit more about that. The water molecule I showed you before is now shown up front, up close and personal. You see the oxygen atom with that lowercase delta with a minus beside it, meaning that it's been pulling electrons not from just one hydrogen, but from two hydrogens, so it has the equivalent of two small partial charges. Each hydrogen has a delta plus, meaning it's only sharing electrons, its own electron with the oxygen, and so it only has a partial positive charge. We note that that is a covalent bond between the hydrogen and the oxygen. And it's covalent because there's not a complete ionization that's happening like happens with sodium and chloride, for example. So there's a sharing, but there's an unequal sharing. Oxygen's holding the electrons closer to it than hydrogen is. So uneven charge distribution is essential, as I said earlier, for hydrogen bonds, understanding all of that. And uneven charge distribution gives rise to polarity. So in this case, we see the molecule is dipolar. It has regions that are more positive and regions that are more negative. That's what a dipole has. It has an uneven distribution of charge. Dipole has an uneven distribution of charge. Because of this uneven distribution of charge, as I said earlier, hydrogen bonds arise. Hydrogen bonds will always involve hydrogen. Now, that may sound like duh, but we forget that. Hydrogen bonds always involve hydrogen. Okay? Hydrogen bonds turn out to be some of the most important bonds in all of biology. When I say biology, I mean biochemistry. They're the most important ones because, as we will see, they're weak enough to be breakable, and they're strong enough to hold things together. And that balance between the weakness and the strength of hydrogen bonds is really, really critical. We'll see this over and over. Okay, a couple more terms. Hydrophilic versus hydrophobic. You probably got this in some of your basic chemistry classes. A molecule that is hydrophilic. Hydrophilic means it likes water. It means it dissolves in water readily. Hydrophobic means it doesn't like water. It doesn't dissolve in water readily. You put oil and water together, they don't mix, they separate, right? So you try to put a hydrophobic substance and dissolve it in a hydrophilic substance, and it ain't going to go. Okay? So we see molecules like hexane, nonpolar hydrocarbons that are hydrophobic. We will see biological molecules like lipids that are hydrophobic. Fats and cholesterol are very hydrophobic. And since our body is mostly water, we've got a real problem when we try to move something like a hydrophobic fat from one part of our body to another. We've got to have some special ways of doing that because it won't just dissolve in the liquid of our bloodstream. Okay. Hydrophilic compounds, of course, are ionic compounds. Anything that is able to ionize or anything that is able to make hydrogen bonds to a reasonable extent will be hydrophilic and will dissolve in water. Ethanol, for example, is an organic compound, But it's hydrophilic because it makes good hydrogen bonds between its OH group and water. So you drink beer, you don't see things separate out. Only your visual field separates things out. Right? Okay. There's my graphic. Okay? It looks like a toilet bowl, but it's actually a little dish that somebody had of oil with droplets of balsamic vinegar in it. Balsamic vinegar being aqueous, oil being hydrophobic. Aqueous compounds, of course, are all hydrophilic. There's another term to know, amphiphilic. And amphiphilic, as its name suggests, is somewhere in between. Somewhere in between. You learn in organic chemistry about what soaps and detergents are, and soaps and detergents are examples of molecules that are amphiphilic, meaning that they have characteristics of hydrophobic molecules and characteristics of hydrophilic molecules. Soaps and detergents have a portion of them that is very nonpolar. The left part of this molecule is very nonpolar. If we only had the left portion of this molecule, it would not dissolve in water. The right portion of this molecule is very hydrophilic. Hydrophobic on the left, hydrophilic on the right. A hydrophilic molecule will associate with water. Well, this guy has characteristics of both. A soap is a soap, and a soap is dissolved in water because that part that is hydrophilic associates with water, and the hydrophobic part, as we will see, associates with itself. That forms what we call a micelle. We can see in this case that the green part, okay, which is the sort of outer part that's also the blue part, they're the same thing. Okay, the green, isn't that kind of odd? The green and the blue are the same thing. The hydrophilic part's on the outside. The hydrophobic parts are all associating with himself on the inside. And the reason you use soap to wash your hands is the grease that is hydrophobic that's on your hands associates with the inner part, right? And the outer part, which is water soluble, just carries it away. So you wash your hands, grease gets carried off of your hands. There's other amphiphilic substances that are very important. We'll talk a lot about these as we get talking about membranes. The membranes of our cells are amphiphilic. They have a portion of them that's polar and a portion of them that's nonpolar. You can see that labeled on the figure on the screen. Okay? The polar part being on the right. Whenever you see a phosphate, you can bet it's polar. It's going to allow a molecule to have associations with water and very likely to interact strongly with water. The nonpolar part on the right, those are typically long-chain fatty acids like you saw in that soap that are very hydrophobic. Now, when we had the soap molecule, we only saw one of those long chains sticking off of there. Here we have two. One's labeled R1, and the other is labeled R2. Now, that may seem like it's a minor thing, and it is a minor thing until we see what happens when they hit water. When this thing hits water, what happens is it'll form a bilayer instead of forming a micelle. That bilayer is what makes our membranes. Examples of membrane lipids include the glycerophospholipids. You also hear them called phosphoglycerides. The sphingolipids are a related set of compounds that I'll talk about. So here's what I was trying to show you. The lipid bilayer looks like this. We see it didn't arrange itself in that spherical structure we saw of the micelle, but instead it's a bilayer. We have a top and a bottom. We see that that top part has on the very top part the hydrophilic portion, and the bottom part has at its very bottom the hydrophilic portion as well. This is the membrane of a cell. The membrane of a cell has the outside of a cell associating with water outside the cell. It has the inner part associating with the cytoplasm of a cell, which is also a water environment. And in between the two, we've got all the nonpolars interacting with each other. That second tail makes a difference. It prevents that formation of that sphere we saw with the micelle. These membranes that we see, these membranous structures, form spontaneously. It doesn't take anything for the cell to construct those. We can make artificial cells or artificial membranous structures called liposomes if we take these things and we shake them all up. And we see here, well, what's the difference here? I've got a spherical structure, but notice it's a bilayer structure, not a monolayer structure. Our cells have a structure like what's on the right. Again, we'll talk more about membranes later. Here's another depiction of the same thing. We see on the inside portion of this molecule, or this bilayer, we see in the inside portion those nonpolar tails that are associating with each other. If you look closely, you'll see some of them have single bonds. Some of them have double bonds. That'll become important for some of the characteristics of the membranes that we talk about. And the outer portions are where we have the phosphates and other polar compounds. And those phosphates and other polar compounds are interacting with water. So if we were to measure the water relevant to this thing, we would expect that the water would be in the highest concentration on the outside ports, and the further we move in, the less water that we would see. And that's exactly the case. Well, that hydrophobicity and hydrophilicity, water attraction, water phobia, has very important considerations for proteins. Very important considerations for proteins. A very simple depiction of that is shown on the screen here. What you see on the left is a polypeptide chain, meaning that it's got multiple amino acids bonded together. And they have different characteristics. We see on the left that we have a structure that we call unfolded. And on the right, we see a structure that we call folded. You can see that structurally they look different. It's the same protein. But as I said, proteins, structure matters. To understand structure, understand function, we have to understand structure. So the structure on the left is different than the one on the right. We might expect they'd have different functions, and that's exactly right. A protein will not function unless it has the proper structure. And the proper structure of a protein arises as a result of the phenomenon you see very simply on the screen called folding. Folding. Now, there's a lot we understand about folding, and there's a bigger amount we don't know about folding, but we do know some of the considerations. And one of the considerations I'm going to show you because it relates to hydrophilic and hydrophobic. In this screen, I've depicted the hydrophobic amino acids in black, and I've depicted the hydrophilic amino acids in yellow. And we can see that what's happened is that this folding process occurred driving the hydrophilic amino acids to the outside of the protein and the hydrophobic amino acids to the inside of the protein. Just like the lipid bilayer, guys. The hydrophobics associated with each other, the hydrophilics associated on the outside, and since this protein is dissolved in water, guess where the hydrophilics are going to be? Okay? It's a very simple idea. It's a very important driving force for folding proteins. Now, we'll see some proteins aren't found in water, and we might predict that they would fold differently, and we'd be right. We'll talk about that later. So water's chemistry affects molecules. That's really critical, especially critical for proteins. There's that hydrogen bonding. I've gone through that a little earlier. I'm not going to repeat that again, but you can see a little closer that hydrogen bonding that's occurring. Hydrogen bonds occur between substances other than just water to water. Biomolecules have a lot of oxygens and hydrogens and carbons, and those don't share electrons very equally. So if I have a water molecule, as you see on the structure on the left, what you can see there then is that that water molecule has a partial negatively charged oxygen. If I have, let's say, ethanol, like I had before, where I have an OH, that H is going to be partly positive. It's going to be attracted to the water, and that's how ethanol dissolves in water. Hydrogen bonds form, as you see here. It doesn't have to be coming from an ethanol. It could be coming, in this case, from an amine. Amino acids have a structure like seen in the middle. And when you look at the structure in the middle, nitrogen's electronegativity is stronger than that of hydrogen. So nitrogen is partly negatively charged. Hydrogen is partly positively charged. And again, the partial positively charged hydrogen is going to be attracted to water. Water's everywhere. Water's 70% of what we are. Similarly, on the right, double bonded oxygen, same issue. Oxygen being more negatively charged because it has a greater electronegativity, attracted to the positively charged hydrogen on a water molecule. A lot of different ways this can happen, okay? Whoop, there we go again. If we look at the structure on the left, okay, we see formation, how ions dissolve in water. And again, ions dissolve in water because of hydrogen bonds. That's how ions are dissolving in water. Hydrogen bonds pull them apart. That positively charged potassium ion is pulled away from its counter ion by the water atoms, or water molecules, rather, as you see here. Hydrogen bonding is very critical, as I said. It's one of the most important forces we see in all of biochemistry. One of the most important forces. Everybody studied in high school, in college, DNA, G pairs with C, A pairs with T. What holds G and C together are hydrogen bonds. What holds A and T together are hydrogen bonds. Your DNA, double helix, is held together, at least partly, by hydrogen bonds. Those are pretty weak forces. Don't mess with your hydrogen bonds, guys. Okay, so I want to put this in perspective for you with respect to hydrogen bonds and covalent bonds. All right? As I said, hydrogen bonds are weak forces. We can see the bond energy associated with covalent bonds varies with the specific type of covalent bond that we're talking about. But we see a bond energy of 460 kilojoules per mole for an OH. That's a strong force. That means we would have to supply that amount of energy to break that bond. If we compare that with the forces holding together a hydrogen bond, we see that hydrogen bonds pop in at about 20 kilojoules per mole. It's a lot easier to break a hydrogen bond than it is to break a covalent bond. Very important. Okay? We can break hydrogen bonds by boiling it. Hydrogen bonds hold together DNA. Hydrogen bonds hold together proteins. We cook our food because we're breaking hydrogen bonds in proteins and DNA of the cells that are in, the bacteria that are in the food that we eat, and therefore they die because their proteins don't function, their DNA can't do anything. They're dead. We've broken their hydrogen bonds. Okay? All right. Now, the main point of what I want to say today is actually right here. Okay? Ionization. Because ionization, as I said, is going to be critical for everybody to understand what's happening with a protein in a solution. I'll talk more about that next week, but today we're just going to go through the basics of ionization. Ionization literally means the formation of an ion. And how do ions form? In biochemistry, ions form by loss or gain of a proton. Loss or gain of a proton. That's basically how ions form in biochemistry. Okay? So all of a sudden, our understanding has gotten very simple. Loss or gain of a proton. A proton has a positive charge. If you gain a positive charge, you're one more charge positive. If you lose a positive charge, you're one less charge positive. If I start with something, like acetic acid, on the very top line, I can see that with the proton on, I have CH3COOH. That is a charge of 0. When I lose the proton that comes off of the carboxyl group, and yes, you should know that, loss of one positive charge means it's one less positive charge charged. So that means that the loss of the proton results in the acetate ion, which has a charge of -1. Each time I lose a proton, I lose one more charge. If I look at oxalic acid, for example, oxalic acid lost its first proton, has a charge of -1. The one below it should have a charge on each end, and I see it doesn't. It should be having a charge on the left end. It has a charge of -2 because it's lost 2 protons. Okay? We can see it with malonic acid quite clearly. If you go down to the third from the bottom, that minus charge is more clear than it is on the one for oxalic acid. But the point is, each time you lose a proton, you lose a positive charge. Each time you gain a proton, you gain a positive charge. Everybody learned in freshman chemistry and memorized in freshman chemistry. pH is the negative log of the hydrogen ion concentration. Everybody, I hope, learned that POH is the negative log of the hydroxide ion concentration. And we can take and we can extend this as far as we want to, and we can say that the P Kevin Ahern is equal to the negative log of Kevin Ahern. So if you could take the negative log of me, you would have the P K Kevin Ahern. All right? Get the idea? All right. We remember that in water, pH plus POH equals 14, because the concentration of hydrogen ions equals the concentration of hydroxide ions in pure water, and they're both 10 to the -7th. So the pH of those two add up to 14. If I increase the pH, I decrease the POH, because they've always got to add up to 14. If the pH is 1, the POH is 13. If the pH is 9, the POH is 5. Pretty simple math. Weak acids dissociate. Now, if there's something every person in here ought to write down or have it embedded in their memory, this is it. And I'll wager you that come exam time, half of you will forget to write this down on your exam. I'll even give it to you on the exam, but I want you to write it down to remind yourself. This is what a weak acid does. And notice I said a weak acid, because that's what we're really concerned with in buffers and in most of biochemistry. Schematically, an acid is something that has protons that it can lose. A salt is something that has lost a proton. So the difference between an acid and a salt, and we're not going to talk about conjugate acids and conjugate bases and all that bullshit, all right? We're going to talk about simple things, acid, salt. An acid will always have one more proton than its salt. Very simple, right? HA has one more proton than A minus. Everybody with me? Don't forget that. Now, what you see on the screen is ionization. An ion is forming if we go to the right. There's actually two ions forming. Okay? You see a salt, which is the A minus, and we see a proton. Okay? The only things we name here are the acid, which is on the left, and notice that the acid is HA. It's not H+. The acid is HA because I said it has one more proton than the salt does. I don't name the proton. What weak acids do is important. It's essential. What weak acids do is they don't lose all their protons unless we drastically change their pH. Don't lose all their protons unless we drastically change the pH. In freshman chemistry, you probably did titrations. And in titrations, you had to add a little bit of, let's say, sodium hydroxide to a solution and then measure the pH. Each time you add a little bit more, and you see the pH going up and up and up and up. And you probably had to plot that, something like what you see here. If you do that for something that partly dissociates, like a weak acid, and by the way, a definition of a weak acid is an acid that does not completely dissociate when you put it in water. A strong acid will completely dissociate when you put it in water. HCl is a strong acid. If I take HCl, I take 50 million molecules of HCl, and I dump 50 million molecules of HCl into water, I will get 50 million molecules of H+, and 50 million molecules of Cl-. If I put 50 million molecules of acetic acid in water, I might get a few thousand molecules of salt, and I would be left with all the rest in the acid form. Understand? There's a big difference. I did not completely dissociate with the weak acid. I completely dissociated with the strong acid. That's a very important point. Weak acids do not completely dissociate. Well, how much do they dissociate? It depends on the pH of the solution we put them into. Depends on the pH of the solution we put them into. As we adjust that pH, we see varying amounts of acid and salt. Let's look at this titration curve. This is a titration curve for carbonic acid. Carbonic acid is an important compound in your blood because it's a way for your body to get rid of carbon dioxide. If we start at a low pH, we have essentially everything in the form of carbonic acid, H2CO3. It has all the protons on. As we start adding sodium hydroxide, we're changing that solution, and protons are coming off. Why are protons coming off? Anybody know why protons are coming off? Anybody? Because what? Well, you're increasing the pH, but why are the protons coming off? Why does increasing the pH make the protons come off? Are there any chemistry people in here? Yes. So he's on the right track. Those excess hydroxyls that hydrogen ions want to combine with. What's the principle? There's a principle that we call that's associated with this. Anybody ever heard of Le Chatelier's principle? What does Le Chatelier's principle tell us? A system at equilibrium, if the equilibrium is disturbed, the system will try to reestablish the equilibrium. If I take a solution that's got a few protons in it and I dump a bunch of hydroxide into it, what happens to those protons? They get gobbled up by the hydroxide, right? Hydroxide plus proton equals water. So if I take protons away from the solution, the solution tries to reestablish the condition it had before, and that's why the carbonic acid gives up protons. Le Chatelier's principle. The more protons I take away, the more protons will be given up by the carbonic acid. Let's imagine I start with 50 million to begin with, and I add 25 million hydroxyls. If I add 25 million hydroxyls, what's going to happen? Well, presumably I'm going to pull off 25 million protons, right? And how many carbonic acids am I going to be left with? I've made a salt, right? Let's back up. I'm going too fast. Back up. I start with H2CO3, right? This is Friday. You guys look like you're dead. What do I got to do? Nobody wants to say. It's a dead group. All right. I've got H2CO3. I take a proton off of it. What am I going to get? HCO3 minus. You can see it on the screen. That's the salt. If I add sodium hydroxide, I'm going to make salt. I'm going to lose acid. I'm going to make salt, right? So if I start with 50 million acids, carbonic acid, H2CO3, I add 25 million hydroxides. 25 million of those H2CO3s are going to give up protons. I'm going to end up with 25 million HCO3 minus and 25 million H2CO3, right? I would have equal amounts of salt and acid, right? Everybody with me? That turns out to be the middle point in this graph. The middle point in the graph, I have equal amounts of salt and acid. Equal. And you'll notice, how is the pH changing in that region? The pH increase is slowing down, right? The least change in pH is occurring at the place where the salt equals the acid. Very important point. The strongest buffering point for a buffer, and we're talking about a buffer here, the strongest buffering point is when salt equals acid. Strongest buffering point is when salt equals acid. Well, now I've introduced the term I didn't define. It's a bad thing for instructor. Bad instructor. Okay? A buffer is a system that resists change in pH. A buffer is a system that resists change in pH. When I say the word buffer, this is something else you're not going to write down on an exam. Half of you will not do this. When I say the word buffer, the first thing that you should think of is salt, and the second thing you should think of is acid. Either order doesn't matter. Buffer equals salt plus acid. Plant that in your head. If I don't have a salt and an acid, I don't have a buffer. HCl does not have a salt and an acid. It's not a buffer. It's all H plus or it's Cl minus. We don't see a buffer. It doesn't exhibit this behavior that you see here. Buffers resist change in pH because they have the ability to donate protons as appropriate. And what if I put protons in the solution? What if I take HCl, which is a pure source of protons, and I'm at that top point in the graph and I start putting HCl in? What's going to happen? Well, the Chatelier's principle says that I'm disturbing the system. The system's going to try to balance that. So it's going to try to take up those extra protons that I'm adding, and I'm going to see salt converted into acid. Right? Simple buffer. Add protons, I make acid. I add hydroxides, I make salt. And there's a one-to-one relationship. If you remember that, you can work 90% of the problems I will throw your direction. You need to know that. Well, this brings us to the equation that I will give you on the first exam. I don't care that you memorize it. I care that you know how to use it. And that is this. It's the HendersonHasselbalch equation. And the HendersonHasselbalch equation says that the pH is equal to the pKa, which I'll define later. That's a constant for an acid. It's a measure of the strength of a weak acid. It's equal to the pKa plus the log, in this case, of HCO3 minus over H2CO3. That is salt over acid. pH, pKa plus AC minus over HAC. In general, pH, there's in big letters. pH equals pKa plus log of the concentration of the salt divided by the concentration of the acid. As the concentration of salt and acid changes, the pH will change. And as the pH changes, so will change the concentration of salts and acids. The only things that can change here are pH, salt, and acid. pKa is a constant. I said pKa is a constant for an acid. It's a measure of the strength of a weak acid. Only weak acids have pKa's. HCl doesn't have a pKa effectively. The lower the pKa, the stronger the weak acid. The higher the pKa, the weaker the weak acid. Stronger, weaker, 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 stronger. Gets a little messy there, right? I think you can follow that. If I give you formic acid, it has a pKa of 3.75, and I gave you acetic acid, which has a pKa of 4.76, and I said, which is a stronger weak acid? I would hope you would tell me that formic acid is the stronger weak acid because it has a lower pKa. Now, in recitation next week, one of the things you're going to start working on are problems related to this, so I'm not going to spend a lot of time going through these. But I will tell you that if you're having problems understanding this, you're concerned or anything, come see me. My office is ALS 2145. I think it's on the class webpage. If it's not, I'll tell you here. ALS 2145. You don't need to make an appointment because my policy is when I'm in my office, when I'm not in a meeting or something, I'm in my office, and you're always welcome to come by. If you want to make an appointment, you can. Did you have a question? Oh, okay. Okay. Buffers resist change in pH. Buffers require weak acid systems. Buffers have capacity. The more acid and salt I have, the more capacity I have. And I can exceed capacity, as we will see. There's the thing that you saw before. Notice that I had 0.5 equivalents of OH, and that's where the point where I had half salt, half acid, because I'd eaten up half of what was there. Rapid pH change away from the buffering region, limited pH change in the region of the buffer. You can assume if you're within 1 pH unit of the pKa, and by the way, the pKa is the pH at which the buffering capacity is strongest. If you're within 1 pH unit of the pKa, you have a buffer. If you're outside that, it doesn't act very well as a buffer. Okay, we'll go through that. We'll talk about that next week. Amino acids with multiple pKa's, multiple things that can ionize. Okay, we're not done yet. Relax. Estimating, we'll talk about that next time. We'll come back to that. Oh boy. And now, to finish today, I've got a song that summarizes all of what I've told you. All right? And you know my deal. Loud singing, we have. Okay. It's called HendersonHasselbalch. Come on. Ah. Oh, woe is me. Here we go. Your PKs can make me lie in bed awake. They give me really bad headaches. Oh, hear my plea. Salt-acid ratios help keep the pH froze by buffering. They show tenacity to complete audacity to fuse with incapacity. I can't hear you. Contain things. Know when H's fly, a buffer will defy them actively. Those protons cannot waltz when they get bound to salts. With this, the chain change in pH halts all praise to thee. Now that I've addressed this topic for the test, I've got to know how the pH I can say equals the pKa in sum with log of S, or I know. All right, guys. Okay.